is something due. There's a lab that's due on Tuesday also. We're doing a blood lab. Let me just check on that day if we possibly can move that over to Thursday, just to make it easier for you guys. And I think there might be a quiz on that Thursday. And so what we're going to do is just, like I said, cover as much as possible so that next Tuesday it's just going to be lecture, so if you want to show up, show up. We'll keep that as short and sweet as possible. And then on next Thursday we'll have a quiz. We'll do the rest of lecture. What's that?
We have a quiz this Thursday?
Next Thursday, this Thursday, 2 days from now, yes, we will have a quiz. And I'll bring that up at the end. Next Thursday will be the quiz, lecture, and then lab. And that we're going to do blood pressures, and we should be set from there. And that'll keep us on track. Okay. There may be some topics when we get to the vessel lecture. I may send you guys just kind of, when we do cover lecture, I may just say you guys can review this on your own time. It's pretty straightforward. Just like I do with pretty much most lectures, just a quick anatomical background before we actually get into the nitty-gritty and we get into the physiology of things. Does that work with everybody? All right. So what we're going to do, we're going to get back to where we started. When we talked last Thursday, we specifically talked about blood. And like I said, that's fairly straightforward. When we get into how hemoglobin works, that is what we're going to be talking about in our vessel lecture. So right now, we're just honing our focus on the heart. So by now, you guys saw this slide. This slide covers the pathways. Again, stuff that you would have covered in anatomy. And we're looking specifically at the pulmonary circuit, systemic circuit, which is okay because we're going to revisit this again in our vessel lecture when we start that at the end of Thursday. If we have time, I believe we should be able to start this on Thursday. We'll talk about the different vessels. Where we want to really hone in our focus on is the heart. So today, we are going to be covering the cells. So we're going to start back at our cellular layer, and we're going to first take a look at how these two types of cells that are prominent in the heart are going to work electrically. So we're going to have to go over all that stuff when we looked at depolarizations, repolarizations, in order to understand the electrical excitability of the cells, and then what the impact is on the cardiac muscle cell. So what we're looking at are the pacemaker cells. These are specialized cells that are attuned to being autorhythmic. And what do we mean by autorhythmic? They, if you were to take a pacemaker cell, put it in a petri dish, give it enough oxygen, give it enough nutrients, those cells will beat, and they'll beat on their own. Okay? And their whole purpose is to just beat. They are the sort of the conductor of the heart. They tell the heart when to fire, and they do it because that's exactly what they do. They're pacemakers of the cells. Now, they don't do anything else but that. They don't contract. They are limited in terms of their contractile property. In fact, if you were to take a look at the image on the bottom, you can see there's some presence of striations, which gives you shows you the ability that these cells can contract if they need to, but those things have been inactivated. Really, their entire purpose is to lead the contraction so that the heart can pump in a uniform fashion. And we'll talk about that as we start to talk more about the electrical excitability. You'll learn more about how the cardiac muscle cells will contract, and then we'll take that overall and take a look at how the heart tissue will beat in this fashion in order to get blood to pump efficiently throughout the heart. So that's the pacemaker cells. The other portion of the cardiac muscle cells, where we're going to again look at the action potentials that are generated in order to result in that muscle contraction that takes place. So we'll take a look at the properties of that fairly soon. So cardiac muscle cells, if we're to take a look at them, again, we didn't spend too much time on our muscles, but the main characteristic of our cardiac muscle cells is that they're branched. You can see the branching that takes place, a lot of different wide branches. They're short, and they're fairly wide cardiac muscle cells. You'll see in the book, they do refer to different names: myocardiocytes, myocardial cells, myocytes of the heart, or simply put, cardiac muscle cells. So the whole, by looking at all of these terms, you can see they all have something in common: myo, cardio, and then cytes, meaning cells. So whether I call it myocardiocytes, I call it cardiac muscle cells, myocardial cells, you get the general idea. These are going to be the contractile cells. Pacemaker cells are going to be called pacemaker cells. So you know exactly what I'm referring to. These are the non-contractile, these are the contractile cells. Looking at the cardiac muscle cells, you can see that on our images to the right, these junctions in between adjacent muscle cells are connected together. And you can see that on our cells over here. You can see these, what are called intercalated discs. Basically, these cells are zippered together. In fact, the way they're zippered together, they have desmosomes. And if you remember from our tissue lecture, desmosomes serve for the purposes of anchoring the cells together. That keeps them stuck together. Now imagine if these cells didn't have anything that anchored them together, these cells are constantly contracting. And over time, they could start to break apart. So we want something that glues them together tightly, and that's exactly what we want. So in between, in these intercalated discs, what you'll also see, aside from the desmosomes, you're going to see these gap junctions. And if you remember, again, from our tissue lecture, the whole purpose of those gap junctions are to allow ions to move through. So what we've been learning about neurons, when neurons communicate with one another, you'll see the electrical activity that takes place within this neuron number one. You see a neurotransmitter diffuse across, that's the chemical portion, and then it makes its way over, that neurotransmitter targets the next neuron, binds to a receptor, causes electrical activity to take place on the second neuron. Here we get direct ions moving from one cell to the next. So if we were to see a wave of depolarization, that is a rush of sodium starting in this one muscle cell, those sodium that make their way out from the extracellular fluid into the intracellular fluid will start to rush through those gap junctions. And that will cause excitation on those adjacent muscle cells. So you'll see how those waves take place, and that's how we can have electrical activity take place very quickly. There's no diffusion across any synapse. It's direct ion transfer, so direct electrical transfer from one cell to the next. Now, the other thing to take a look at the cardiac muscle cells is that we have high mitochondrial density. And by that, we know exactly what the mitochondria is responsible for. We know it's for the generation of ATP. We know it's going to undergo glycolysis. It's going to use those mitochondria in order to generate that ATP efficiently. So therefore, it's not really using anaerobic. It's not primarily dependent on anaerobic metabolism, i.e. glycolysis. It needs to undergo the Krebs cycle, undergo the electron transport chain in order to efficiently make energy. The other thing too is what, and this is covered in the book, but not really important for the purpose of the lecture. The heart doesn't only use glucose, it can also use lactic acid as well as a byproduct. Therefore, it does efficiently, obviously it's a critical tissue. It needs to work, and it needs to generate ATP to continually pump blood through. So it's going to use any metabolic substrates it can in order to generate optimal amounts of ATP. Now, I did mention earlier, on the earlier slide, you saw prolonged depolarization. That's the main characteristic of our muscle cells that you'll be able to see when we get into our membrane potential, the changes in the membrane potential on our later slide. I know in all of our neurophysiology lecture, when we talked about our neurons, we talked about sodium and we talked about potassium and the movement of those ions. The third thing we're going to see that's characteristic in our cardiac muscle cells are the calcium channels. So we're going to see how these voltage-gated calcium channels are going to be involved in prolonging that depolarization. Looking at the cardiac muscle fiber arrangement, here's just an interesting slide behind it, just graphically showing you how all the muscle fibers, you're going to see our actin and myosin, which you learned in anatomy, are all creating these cross bridges to basically allow the contractile tissue, the cardiac muscle cells, to contract one another. So here you can see the different fibers, the different fibrous proteins that create bridges together. And I'll be able to show you as we go through the generation of tension that takes place. At the start of depolarization is going to start to create these bridges that are pulling one another and effectively sliding. So here you see the sliding filament theory in order to shorten the muscle fibers that you see on our prior slide, in order to shorten these but to also elicit contraction. So keep that in mind when we start looking at not just that individual muscle cell, but how all these cells operate together to create contraction that takes place in that tissue. Now here we have an exercise. So what I want to point out in this exercise is that while we have here these hexagons are a bunch of muscle cells, cardiac muscle cells. And in between here, you'll see the gap junctions. Each of these represent gap junctions. What we have are just and you can just think of just a blanket sheet of a bunch of cardiac muscle cells. They're all arranged together, and they're all held together by their intercalated discs with gap junctions that allow the transfer of ions in and out. And so take these muscle cells by themselves, and like I said earlier, put them in a dish, give them enough oxygen, give them enough glucose, and they'll beat on their own. And they'll continually beat as long as you provide enough nutrients for them. Now, each of these by themselves, these cardiac muscle cells, beat about 20 to 30 beats per minute, and you'll be able to see that when we talk about that in a later slide. If left on their own, 20 to 30 beats per minute, and it's not like individual cells are beating at their own rhythm. What's going to take place is you're going to take one cell, and one cell that starts to depolarize. You remember in our action potential, the first phase of the action potential is what? You guys tell me. What ion is rushing into the cell? Sodium, right? So we're going to get sodium, extracellular sodium, and I'm just going to start. I'm going to pick on this this cell right here, and we're going to get sodium rushing in. That's going to be the first phase of the action potential. And that sodium that rushes in is going to start moving through those gap junctions. So while this cell depolarizes and undergoes that first wave, those sodium ions are going to start moving over, and this is also going to depolarize nearby adjacent muscle cells. So as this starts the wave, then more sodium comes in here, more sodium comes in here, this depolarizes, brings this to threshold. And as this starts to reach threshold and causes more sodium to rush in, all the other ones coming surrounding will also start to depolarize. So what starts off as this cell firing creates waves that propagate away from that single point. Now take this muscle cell, strip it of all of its contractile proteins. The whole sole purpose of this is now that if this beats at 20, 30 beats per minute, all of these will beat at 20, 30 beats per minute. So this is going to start that point at which contractions, at least when our depolarizations will take place. So this cell, we want to reprogram. We want to remove the contractile proteins, and we're going to tell this cell to beat at about 100 times per minute. That means even though the other cells can beat at 20, 30 beats per minute, every time this fires, everybody else fires. So this reprogram is exactly what happens with what's called our pacemaker cells. These pacemaker cells are geared to contract and be the pacemaker cells. They're the ones that lead when the rest of the nearby cells will fire. So looking at these pacemaker cells, like I said, they're non-contractile. They have the ability to spontaneously depolarize. So they beat at 100 beats per minute, and they tell when everybody else beats.
So those are pacemaker cells. Earlier you said they were cardiac.
Okay, so all of these are cardiac muscle cells.
Okay.
I'm just pinpointing this one and saying this is the one that's going to be the one that's most excitable.
Okay.
If you reprogram it, and that's basically what's happening. When you look at cardiac stem cells, they can either be pacemaker cells or they can be muscle cells. And these cells are going to be the one that's reprogrammed to be only the beating cell. Okay, everybody else is going to contract. They do the same thing. You're going to see the same action potentials, but there's going to be a slight difference in those action potentials, and that's what I'm going to go into in a little bit. So these cells are specifically the pacemaker cells. And if you were to wrap this layer over a heart, you can see that there's going to be a specific area that will beat at 100 beats per minute, a specific group of cells called the SA node. And that SA node, which we'll discuss later, is called the sinoatrial node. And as you can surmise from the name, it's going to be located towards the atria, and I'll show you that on our next slide. So these SA node cells are pacemaker cells. They don't contract. All they do is they spontaneously depolarize. this is the term which we refer to as autorhythmicity, meaning automatic rhythm. And as these fire, this is going to create the rush of sodium ions that move all the way through. Every time this beats, sodium rushes in. Sodium is going to move through those gap junctions and travel down all the branched cardiac muscle cells that they innervate. So let's take a look on the next slide over. What we're going to do is we're going to talk about what that membrane potential looks like. So here we're going to first look at, these are going to be our pacemaker cells. And I've got this all drawn out for you on the next couple slides, so don't worry about having to draw for accuracy. I'm going to be referring to each of these steps below. So if you want to just kind of listen through, we can start from there. Now you'll see that most of the cells that we're going to start off with, you'll see our threshold that's drawn in blue. We're going to start off at about negative 60 millivolts as our resting membrane potential. These are our pacemaker cells that are at rest. Same principles apply as to why it's negative 60, negative 70. Again, large anions inside the cell. You've got our sodium-potassium pumps, and you have leaky potassium channels that also contribute to why it's negative 60 as resting membrane potential. And what we're going to start off with, again, is at our starting point right at 0 seconds. And we're going to watch how this pacemaker responds over time. Now the critical thing that you're going to be able to see about our sodium channels, and you can see this on our first slide over, the very beginning is the presence of leaky sodium channels. Now think about what happens. And I know I mentioned leaky potassium channels. Those are still there, but we have more prominent leaky sodium channels. So what does that mean? We've got a cell on the outside. We've got leaky sodium channels on the cell membrane itself. That means when our cell is at rest, we've got a high concentration gradient of sodium on the outside wanting to be driven through the inside. So this means as the cell is at rest, we're going to see sodium going in. And as sodium goes in, we know that that's going to be decreasing the membrane potential. And when I mean decrease, it means bring it up towards 0. So there's going to be an upswing. And so it's going to just slowly, based on the number of sodium channels open, will allow this continual rise in the membrane potential, up to the point that it reaches negative 50. So by just virtue of the number of sodium channels open, this will continually rise and rise and rise. But right at the point at negative 50 is we're going to notice this point, we're going to see a huge onslaught of a depolarization curve. So what started off as this initial portion is sodium entering through the leaky channels. This second portion, this next upswing, is going to be calcium channels. So calcium channels opening at that point. Our voltage-gated calcium channels will now open up, and that's going to allow now calcium, which is high extracellularly compared to that intracellularly. We have calcium rushing in, and that's going to further depolarize. So now we see the added effect of calcium, a positive ion. We see positive sodium ions coming in. We see now a huge rush of calcium, positive ions coming in as well, because we've opened up those voltage-gated channels at threshold. Once we get to the very top, and we're going to get closer to about 0 to about 10 millivolts, right up at that very top, you're going to see calcium channels close. And then we're going to start to see potassium channels open up. And knowing that potassium is leaving the cell, again, high concentration, the gradient on the inside, wanting to leave the cell, our voltage-gated potassium channels open, and then we're going to repolarize. And that's going to keep repolarizing We're going to get to the point where we repolarize close to resting membrane potential, and then we start back at where we started. Right at this point, our potassium channels close, and we start again. Again, we're back at resting membrane potential. Our leaky channels are always open, and then it takes us back to threshold. At threshold, calcium channels open, rush calcium in, potassium out, back to resting membrane potential. Okay. This slope right here that I'm referring to is what we call our pacemaker potential. And the rate of that pacemaker potential, how fast do we start from resting membrane potential all the way to threshold, is dependent on the number of sodium channels open. So we'll look at that. If I were to open up more channels, you would notice that the rate, and let me draw this in blue. If I increase the rate at which our pacemaker potential reaches threshold, you'll see that the action potential will take place even faster. And each time we have an action potential take place is a heartbeat. Like I said, it's a pacemaker that's going to start that wave of depolarizations for those adjacent cells. So in that portion where you saw 1 in red, our pacemaker's lower rate of change in our pacemaker potential, then we see 1 action potential take place. Here you're able to see 2 because we see an increased rate. So this difference, everything below threshold, our pacemaker potential is what's going to determine when that action potential takes place, and therefore that wave of depolarization for the rest of the muscle cells. What questions do we have about this?
So what if pacemaker potential is just basically
The rate at which we reach threshold. It's,
because you're opening up what channel?
yeah, you're opening up more sodium channels.
Okay.
Yeah. So the more sodium channels that are opened, that means that's the rate at which we reach threshold. So more sodium going in brings us more and more and more towards depolarization, and eventually we reach threshold, we open up those calcium channels.
So it doesn't matter if you increase the calcium channels too?
Because once we open up those voltage-gated calcium channels, then you're going to undergo that whole rhythm. You're going to undergo the depolarization to repolarization. So this is where calcium comes into play. What you learned last time was the action potential where sodium rushes in and potassium comes out. Calcium just comes in to add to the pacemaker potential to really cause that deflection. This is going to be the huge wave at which once this takes place, once we get this action potential that takes place, that constitutes 1 heartbeat. Then we're going to go back, and then the rate at which we reach threshold again is going to see the number of waveforms that take place. That answer your questions? So looking at all of these will basically show you, again, everything that I've drawn. Potassium channels closing, slow sodium channels opening, sodium entering, calcium, potassium, again. All of that leads to our action potential. And again, the next slide over breaks that down for you. So in reality, we don't really see a resting membrane potential that's stable. And that's all due to those leaky sodium channels. Anytime you have a leaky channel open, it's just going to allow things to move down its gradient. There's nothing regulating those ions or preventing those ions from coming through. And for calcium, however, on the other hand, those voltage-gated channels, we really need the right voltage in order to trigger those gates to open, to trigger an action potential to take place. So like I said, in that image earlier where I had drawn out all the heart cells, the area that's going to be the most sensitive, that's going to lead the pacemaker cells, are going to be the sinoatrial node. And the sinoatrial node is actually a very small area. It's about, I believe, like 2 to 3 millimeters of an area of concentrated pacemaker cells. And like I said, they beat on their own. Give them enough nutrients, and they will beat. And dependent on that pacemaker potential is the rate at which those sinoatrial node, which contains those pacemaker cells, will depolarize. And this, if you were to count the number of beats, or how long the beat takes, you'll see this is about 0.8 seconds. So 60 seconds divided by 0.8, that's not exactly 100. We'll just pretend it's about 100. It's about 100 beats per minute. What we'll see on here, you'll see no CNS innervation, no central nervous system innervation necessary for depolarization, which is true, that if you were to take a heart, and I don't know if you guys ever played like Mortal Kombat from way back in the days. They had that one character, I can't remember who, but they would like reach in, grab the heart. That heart was still beating. Actually, a heart would still technically do that, because it doesn't need the central nervous system for it to beat on its own. Those sinoatrial node contains those pacemaker cells that will beat and still cause the heart to beat on its own. Eventually, it's going to run out of nutrients, and it's going to run out of O2 delivery to that area. Those cells will fail, and then the rest of the other cells will fail as well. So it won't continue indefinitely. That's where it needs blood coming in from the coronary arteries coming in and feeding the rest of the heart. And as I said, the SA node doesn't fire on its own. It doesn't contract. It fires on its own, but it doesn't have any contractile properties. Now, the sinoatrial node, the SA node, isn't the only place that contains pacemaker cells. In fact, the entire conduction system of the heart, which I'll describe in a little bit, also contains pacemaker cells. So I mentioned earlier, this is the SA node, but everything else that you see in red right here, there's a whole highway, a conduction highway, that travels from the atrium down the interventricular septum, which is going to be that muscle mass in between that separates the right from the left ventricles. And then it's going to wrap down to the apex of the heart and travel back up, called the Purkinje fibers. And that's all described for you on the next slide. The conduction system, again, consists of all pacemaker cells that can also fire should any of the prior nodes stop firing. So let me pause right there and let me first describe the entire highway on its own. So sinoatrial node fires about 100 beats per minute. That means every 0.6 seconds that the SA node will fire. And that wave of depolarization travels to all the adjacent muscle cells nearby. And eventually, through those gap junctions, you're going to reach the next node over. And that node is about 0.3 millimeters away. 0.3 millimeters away from one another. The next node is called the AV node, which is called the atrioventricular node. And it's located really at the most posterior portion of the right atrium. This node right here, like I said, about 0.3 millimeters, very small delay, creates this delay that travels from the SA node over to the AV node. Once that depolarization wave makes its way down to the AV node, then it's going to move it's going to travel all the way down what's called the bundle of His. And again, that separates the right ventricle from the left ventricle. That bundle of His will then move down the left and right bundle branches where it'll separate. You've got the left bundle branch, and that's going to travel down the interventricular septum, down to the Purkinje fibers. Purkinje fibers are the ones that wrap around, and then it makes its way to the outer, the lateral sides of the heart. So looking at this wave of conduction, again, to start off, sinoatrial node sends its wave over. There's a slight delay by the time I get over to the AV node. AV node now travels down this way, down the bundle of His, down the bundle branches, left and right bundle branches, and then wraps around. Okay. So that conduction pathway, yes?
Sorry, can you do that one more time?
So SA node fires every 0.6 seconds, delayed as it travels over to the AV node. AV node then fires as a result of that depolarization wave. Again, that depolarization wave is the wave of sodium rushing in. Once these fire, this is going to send that pathway down the bundle of His, where it's going to separate down the left and right bundle branches embedded within the interventricular septum. And then, once it wraps up, it's going to move towards the apex of the heart, where it wraps around to the Purkinje fibers around. So if I were to, again, for lack of a better analogy, kind of light a match right here, this would travel down this pathway. Okay? It doesn't all fire at the same time. We want it to work like a fuse. Now, the reason we want that to take place is because we want the atria to contract first. Contract atria first, and we'll go over this muscle action that's taking place. Atria want to contract first while the ventricles relax, and as that pathway travels down the interventricular septum, then the atria are going to relax, and then the ventricles are going to squeeze. So that delay from the atria to the ventricles is absolutely necessary in order to allow ejection of blood that's contained within the atria to push down into the ventricles. We want enough time for that to take place. So there's that reason that there's a distance between the SA node as well as the AV node. Now, looking at each of these, like I said, the SA node fires at 100 beats per minute. And if there is a block at the SA node, and you guys will see that on your labs that you guys are going to be doing today. And the labs actually are going to be a take-home lab. You guys can do them on your own. Do we even really talk about blocks in here? Nope, I took that all out. You're really just going to be counting the number of waveforms that take place. If a block were to take place in the SA node, the AV node can fire, and that fires at about 60 to 80 beats per minute. If the AV node were to be blocked, the SA node doesn't fire, the AV node doesn't fire, if you were to cut at any point right there, you would see that the bundle of His would also fire as well. And that's about 40 to about 60 beats per minute. And then lastly, the Purkinje fibers beat at 20 to 40 beats per minute. The Purkinje fibers, down by the ventricles, again, if you were to cut all that off, and the pacemaker cells at the SA node can't communicate down, the AV node can't communicate down, the bundle of His can't communicate down, that means the Purkinje fibers can still beat on their own, but at 20 to 40 beats per minute. So at any point of a block down that conduction pathway, there's still a fail-safe. SA node block, AV node will fire at 60 to 80 beats per minute. If the AV node stops and there's some disruption taking place, we know that the ventricles can still contract and keep blood pumping out of the ventricles. And I'll talk about those blocks later on. Now, what that means, and typically they would see if, let's say, the SA node isn't firing anymore, that means what they can do is they can actually install a pacemaker. And a pacemaker is something that basically is battery operated, and it sends a wire over to that general area. And that's going to stimulate. All it does is give a minor shock that creates the depolarization that results in depolarization taking place. And as long as you can get that signal to start in that area, then it'll travel down and move down the conduction pathway. And all that is, like I said, all it is just an electrical wire into that area. It's probably a lot more complicated than that. But that's going to start that, jumpstart those cells to beat at their own. Another things that they're also doing with these pacemakers is they're trying to find something a little less invasive. And so they're working on what are called bio-pacemaker cells. That means they can take cardiac stem cells. If they have your cardiac stem cells, they can take those stem cells and basically perform some sort of form of genetic expression to create the pacemaker current. And what that involves for that pacemaker current is something that will exhibit that autorhythmicity from resting membrane potential that will lead us to threshold. So really what that comes down to are the expression of those leaky sodium channels. If they can get them embedded around that cell, what am I looking at here? If I can get those, in each of these cells, I can get those expressed leaky sodium channels, they will exhibit that pacemaker potential and then therefore exhibit that autorhythmicity. Again, very simplified version of what they're doing, but much more complicated than that. But they can just basically inject your own cells to become pacemaker cells. That's what it comes down to. What questions do we have about the conduction system, the pacemaker cells and the conduction system? Everybody good? So now that we've talked about the pacemaker cells, the non-contractile autorhythmic cells, let's take a look at the action potential generated by our cardiac muscle cells, our myocardiocytes. Entirely, well, not entirely, but a different action potential of what you saw earlier. The resting membrane potential will start off at -90. And the action potential is going to basically be working the same way that you saw earlier. You'll see the rush of sodium causing depolarization, and then you're going to see repolarization due to potassium. That's not new. We've talked about that already. But what's going to create a prolonged depolarization is due to calcium. The movement of calcium. Just like you saw in our pacemaker cells, calcium is going to serve to prolong that contraction. And let's take a look and see what that looks like. Here you can see the different phases. We're starting off at -90. -90 is when we're going to see sodium, voltage-gated sodium channels open. You're going to see that rush. That's going to take us from -90 all the way up to about +20. And for the purposes of the test, I'm not going to have you guys memorize +20 or how high it goes up. Really, I just want you guys to get the idea that this is the depolarization. This is phase number 1 of the depolarization for a cardiac muscle cell. Once we get to the very top, our voltage-gated sodium channels will close. And then our voltage-gated potassium channels will start to open at this point. Voltage-gated, close, potassium opens. Now we know in a cell, when sodium rushes in, that's going to depolarize, hence bring us that upswing. We know that the other side, when we get potassium rushing out, would be repolarization. Positive ion leaving a positive interior, that's going to make it more negative on the inside. Now what's taking place right up at the middle is calcium. So at this point, right when our voltage-gated sodium channels close, we're going to now open up our voltage-gated calcium channels. So while at that point where you're going to have some potassium leaving, which would repolarize, you're going to also have calcium entering right at the same time. So I've got a positive ion moving out, I got a positive ion moving in. So while potassium efflux is taking place, we get calcium influx. And that should keep us right at that same membrane potential. So therefore, there's no changes taking place from this point up to this point. We keep that cell, that cardiac muscle cell, at a depolarized state. So the length of depolarization is this entire green section right here. We'll talk about why the depolarization is necessary in just a second. Right when we get up to this point, we'll see those calcium channels start to close out, and then continued potassium efflux. So potassium continues to flow out, therefore contributing to the repolarization state. So all in all, what am I getting out of this? I'm getting an extended depolarization state. Everything from this point all the way up to this point. That gives us one thing on here. The next line is to talk about the refractory period. Now, if you remember from our neurophysiology lecture, the refractory period is at the time at which our cardiac, or in our neurons can't fire if there's a second stimulus. Okay. That should mean that if the SA node fires, and then all the cardiac muscle cells nearby fire as well, that means if another one were to fire again for some reason, if we're to fire again, this cardiac muscle cell won't fire a second time because it's already firing. It's already maintained its depolarization due to calcium. So at no point, at this point, at this point, at this point, or at that point, will that cardiac muscle fiber fire again. Why? Because it's already contracting. And it's going to maintain that contraction right at that point in time. So how long is that length of contraction? It takes place from the start of depolarization. We get up to this point right here, and now we're going to maintain that long depolarization state, leading to this whole state of contraction. Now think about this for a second, and I want to go into, now I'm going to be, whoa. Hold on a second. I'll get to that later. I want to kind of fast forward to this slide over, and it's probably about 15 slides over. Action potential and cardiomyocyte contraction. I just moved this slide back a couple slides over after printing what you guys have. So I want to even move this back even further back. Because this is going to show you the action potential that's taking place for that same cardiac muscle cell. Did you guys find this slide?
Yeah.
Now what that's showing you is this extended phase right here. So during this period from over here, from at 0 seconds, all the way out to about 170 seconds. This entire point is the length of the contraction that's taking place. And you can see what's taking place for the purple line representing the tension that takes place with the muscle cell. So what's taking place at the start of depolarization of the nearby, of the muscle cells? They start the contraction, and as they continually contract, they're going to start using those muscle bridges. The myosin heads are going to start pulling against each other and generating enough tension. So it wants to continually increase in the amount of tension up to the point that we get to the very peak. And at that peak is the peak tension development right at that point. Once we reach repolarization, which takes place from this point on, is when the muscle starts to relax and those bridges start to relax on themselves and the tension decreases over and over again. So what this comes out to, and what you learn in our neurophysiology lecture, in order to elicit this muscle tension at the very bottom, we had to have a whole series of action potentials that kept taking place. This is what we called our tetanus. And tetanus results in continued contractions up until the point that we can continue, up to the point we get to the very top. And then, once we decrease the number of action potentials, then we start to see tension decrease. And that was my example that I used earlier when I used the example of the bicep curl. We want to be able to generate enough tension all throughout the length of that state of depolarization, which we're seeing over here, in order to continually generate tension. Why are we doing that? If I were to take a look at a muscle cell and I had one single action potential, that would mean a cell just does this and that's it. That's not what we want. We want to be able to use the heart. The cardiac muscle cells want to be able to squeeze and maintain squeezing. And maintain squeezing for this entire duration in order to develop enough tension, get those muscle cells to continually tighten up and squeeze enough to be able to push blood from one chamber to the next. Whether we're moving from the atria to the ventricles, or from the ventricles to the aorta or the pulmonary trunk. In essence, what we're going to be seeing the muscle do is we're going to want to be able to squeeze long enough to generate enough tension to overcome the pressure in the next chamber. And that will illustrate for you after this. We'll go into the next. We'll talk about the pressures, and I believe we'll talk more about that maybe at the end of today or possibly Thursday. We want to be able to generate enough pressure to be able to push blood over to the next chamber. So I'm going to go back a little bit and take us back to the first time when we started off on. You'll see the length of contraction. And the term contraction is also referred to as systole. I've got other slides that will be using that term, but when you see the term systole, That is contraction. The other state, everything else around here, up until the point we get to the next depolarization, this whole state right here is called, well, it's relaxation. But the technical term for it is called diastole. And now, why do we want an extended period of time for relaxation? If we can answer the question of why do we want a long contraction, was to generate enough tension to be able to raise the pressure in that chamber and get it to squeeze out to the next. And the example I can use is a toothpaste tube. If I want to be able to squeeze that toothpaste out of that toothpaste tube, I got to generate enough pressure on the inside to be able to exceed the pressure on the outside air pressure. So once I'm able to squeeze, now that toothpaste is able to move into the airspace or whatever I'm trying to dump it out into. Now, what I'm going to do next after the end of contraction is I'm going to relax. Because now if I can relax that, I guess toothpaste doesn't work this way, but if you were to relax anything else, what that's going to allow the atria or the ventricles to do, depending on which tissue we're looking at, we're going to allow that to relax. And during that relaxation phase, we're going to have the prior chamber that's going to undergo squeezing that will allow that chamber to fill up as well. I know I'm starting to add a lot more. I'm adding the atria and ventricles, but really I just want you guys to get the idea. Contraction for that muscle, and then relaxation. There's a pattern that cell is going to undergo contraction to relaxation, contraction to relaxation. And that's going to give us enough time for filling or emptying. So with that, what questions do we have about the cardiac muscle action potential?
So this one was cardiac, the other one was pacemaker.
Yeah,
right?
pacemaker for the other. The pacemaker exhibits autorhythmicity. This cardiac muscle action potential, even though it could beat on, it can beat on its own, it's mostly geared for this extended depolarization. And all of it comes down to is where that calcium is involved. In our prior, calcium creates this huge depolarization curve in the very beginning. Sodium is first initially involved, then calcium, then potassium. In our muscle, sodium, voltage-gated sodium channels, voltage-gated calcium channels, then potassium. So be able to, when you're doing your studying, is to think about the movement of the ions and what that's going to do for our membrane, the changes in membrane potential. And that should help explain. And all that stuff that I just drew out here, the movement, think about the movement, think about the potential, the diffusion gradient, the concentration gradient from extracellular to intracellular, and that will help play this all out for you. So why do I bring this up? This whole slide of cardioplegia. Cardioplegia is basically, by breaking up the terms, cardio, meaning heart, plegia, like paralysis, is all talking about how we stop the heart. Now you might be thinking, why do we want to stop the heart? Well, one of the reasons they want to stop the heart, if they're going to perform any open heart surgery. Imagine if you were a surgeon trying to perform surgery on a heart that's beating in front of you. Not really going to be the most optimal way. The proper way is to actually stop the heart. And the way they do that, and I've got a video that talks about the organ care system, specifically for the heart. What they're trying to do in cardioplegia is to get it to stop beating. And the way they can do that is to basically cool down the heart itself. They want to, and why would they want to cool down the heart? Again, it's related to temperature. If you cool a tissue down, that's going to slow the movement of ions. Remember what reducing temperature is going to do. If I'm going to heat something up, that's going to get our ions to move even faster. We're going to increase the amount of kinetic energy. If I cool something down, it's going to slow that down and reduce the amount of kinetic energy. So as ions start to move slower, that technically means that the heart is going to start beating a little bit slower. But what they're also going to be doing is they're going to actually inject a solution that's high in potassium. Typically they use as potassium chloride. And they can inject that into the heart, or they can inject it, they can bathe the heart in potassium chloride, and all that's doing for the cells is increasing the amount of calcium extracellularly. If you want to think about what that mechanic does, well, that was one of your extra credit questions from your quiz, right? What happens if I increase the amount of potassium extracellularly? Now we have to think about what that's going to do. Inside the cell, we already know that there's already a high potassium on the inside. And there should be low potassium on the outside. So with that, there should be an established concentration gradient that pushes outward due to the concentration gradient out. And then the voltage gradient should be inward. So this is concentration gradient, this is voltage gradient going inwards. And again, if you don't remember that, revisit that on your notes. Voltage gradient, because we have a positive ion, it's negative on the inside. Here it's about negative 90 millivolts. We've got a positive ion wanting to go in. We've got potassium wanting to go out. Now, if I increase the amount of potassium on the outside, that's going to decrease the concentration gradient of potassium going out. What that ultimately means, if I got less of a drive to push potassium out, I've got more positive ions on the inside. Hence, what that's going to do for the membrane potential, the resting membrane potential, if it's negative 90, that's going to bring us closer to threshold. So that's going to mean it's going to be more likely to fire. And that means once that heart undergoes that depolarization, it's going to take that one last beat, and then, because I've got more sodium channels activated, it's going to be deactivated, it's going to be inactivated at that point in time. It can't fire again. So it's going to undergo one final beat, and then it's going to stop. So this is our effective way of stopping the heart at that point in time, right after its last beat. So while they stop this, they're not going to just stop it without doing anything else. They're going to now hook up prior to all this, they'll hook up a cardiopulmonary bypass. And basically what it is the heart-lung machine. It's going to take the output of the heart, okay, which would be the aorta, and then they're going to want to also take the output of all the, they'll want to take all the input, mainly our vena cavas, that are returning blood from the systemic circuit, feed that into our bypass machine, pump it back out, be able to oxygenate it, and put it back into the aorta. So this machine is doing what the heart and the lungs are supposed to be doing, pump blood through and oxygenate it in the lungs. Therefore, bypassing the heart. Once you bypass the heart, you can now cool down the tissue, you can do your cold infusion into the arteries to slow it down, and then inject your potassium to give it its final beat. And at that point in time, that heart is now stable, it's not beating anymore. You've stopped the contractile properties, and you can now operate on it. You can cut it open, you can install some valves on there. If any of them are malfunctioning, you can do a heart transplant if you need to, move that malfunctioning heart over, and bring a new heart in. And when you look at heart transplants, all it is plumbing. All they're doing is attaching the vena cavas back in, they're attaching the pulmonary trunk back in, attach the aorta back in, and effectively, that's really it. You're going to you're going to remove the potassium on the outside, you're going to start to heat up the tissue, you're going to start to allow blood to flow back in, and eventually the heart starts working back again. What does that look like? Well, I've got another video, I got a video for you that's going to go over the next, please tell me it's going to work. Yes, great. Not entirely gory or anything, you're just going to see a heart beating. This is what's called the what's called Heart in a Box, and it's made by a company called Transmedics. Now this video is back in, I believe, 2012, so at this time they were doing research on ways to improve the reliability of transporting a heart over from a donor to a recipient. So as you know, sometimes let's say somebody ends up donating the heart, something happens, they're able to strip the organs. You've got a heart that is taken out of a body, and you've got to be able to transport that about 4 or 5 hours, 6 hours over to the next destination and implant that into the recipient in order to make sure that that heart still stays alive. So conventionally, what do they do for heart transplant? They put that heart in ice, and they can slow it down, and typically that should be long enough to be able to get to the point where they can perform the transplant. Now, what this machine does is, again, a company called Transmedics created this machine where you hook up a heart to a huge machine. It's going to feed blood into the system. It's going to keep the heart pumping. Normally, you don't want to transport a beating heart because obviously you don't have enough oxygen, you don't have enough glucose, you don't have any metabolic substrates. That tissue will die on its own. Here, we just want to keep the heart alive by providing enough blood for it. Let me just play the video for you guys. You don't have to watch the entire 9 minutes. Let's see if I can get the audio up and running.
We've all seen it on TV. When a donor heart becomes available, medical teams must move quickly. The organ is removed and preserved by placing it in ice.
No, that's not the one I wanted. One second.
In a cooler full of ice. Here at the United States,
You'll recognize it. This is the very front of UCLA.
Advances in the techniques and technology used, there's one aspect of the process that has pretty much remained the same. Yep. Getting the heart from point A to point B still involves putting it in a cooler full of ice. Here at the UCLA Medical Center, they're testing out a new device that will revolutionize how organs get transported and make a cooler a thing of the past, at least for organ transport. The machine is called the Organ Care System, and it could double the amount of time a heart can remain outside of a body.
A human organ has never been kept alive outside of a human body until this machine became a clinical reality.
Dr. Abbas Ardahali is the head of UCLA's heart and lung transplant program.
It makes intuitive sense to a layperson to say, instead of having my heart on ice, I want it to be warm, I want it to be beating.
For some of his patients, like 44-year-old Sandra Aguilar, this clinical trial isn't just an opportunity to test a cool new device. It's a second chance at life.
También ustedes, y tú sabes que tu hermana son mi pedazo de corazón.
Next. I'm not harmless or anything. I just want to. It's a little corny. She's like, "You are the heart of my life." All right.
Pues mi vida, se puede decir, ha cambiado al
Yeah, it
100%.
decreased quality of life.
Ya no puede trabajar.
Can't work.
Organ Care System, how have they responded to that?
Once they fully understand the concept, they say, so why didn't you guys come up with this sooner?
Today, 8 out of 10 available donor hearts never make it to a patient in need due to a variety of factors, including the distance between hospitals. This device could potentially expand the window of opportunity for transplants while preserving the hearts in a more optimal environment.
We feel very comfortable or confident in the organ care system's ability to at least double the heart transplant volume over the next 5 years.
Waleed Hassanin is the founder and CEO of Transmedics, the company that came up with the organ care system. His eureka moment came while training to be a cardiac surgeon.
The same organ that I'm supposed to be dedicating 10 or 11 years of my life to train how to protect. But when it comes to transplant, which is the pinnacle of cardiac surgery, the heart is just thrown in a plastic bag filled and stored on ice. That was the first revelation that there has to be something better.
Waleed hopes that one day, learning how to use this device will become part of the standard training for surgeons. But for now, the company operates a lab where doctors practice using the organ care system with the closest thing to human hearts, pig hearts. Hopefully they're gonna let me handle the hearts. I don't know if they're gonna trust me. I'm no surgeon, but I've dissected my fair share of animals with my biology background. So I'm gonna do what I can to be able to poke that thing and feel a beating heart in the machine. Once removed from the donor's body, the heart is quickly connected to the device through the aorta. seriously just one of the most amazing things I've ever seen, you guys. There's a heart beating right in front of me. It continues to receive warm blood, as well as a proprietary formula of nutrients. Conceivably, how long could you keep a heart beating on this?
If you can renew the pool,
blood?
it can go as long as you would like.
Tamara Hayal, the VP of Clinical Development at Transmedics, showed me how the device allows clinicians to monitor the heart's rhythm and other vital signs every step of the way. So this thing lets you know that everything is going okay?
Absolutely.
And what happens if something goes wrong?
You'll have detection here and alarms that are going on to tell you that you're out of range, and you can immediately control it. You can change pump flow, you can navigate into menus to change the settings to actually treat this heart. So you have a very comprehensive analysis of how this heart is behaving, and you have a chance to improve that behavior if needed and when needed.
So I have two questions for you. One, how fragile is the heart? And two, can I touch it?
One, it's not very fragile. Two, yes, you can touch it, but it's not the norm during clinical practice.
Okay, not the norm? You're gonna make an
by all
exception?
means, you can touch it. This is a training part today, so by all means, if you want to feel the ventricle pulsating, right side and left side, and you can feel the vessel itself, see the pressure in the vessel.
It's warm.
It's warm, it's beating, it's functioning. It's just functioning as if it's in your body.
And it's not just hearts which can be transported on this system. There's a similar study currently underway with lungs, which has yielded some promising early results.
You see it opening and going up and down?
So far, it's cut the complication rate by more than 60%.
We'll save the lungs for next time. Now that you see how that's working. What we described so far, the pacemaker cells, the action potential is related to the pacemakers, as we mentioned, the SA node, over to the AV node, down the conduction system, down the interventricular septum, the Purkinje fibers wrapping around. Now that we described the muscle cells and how they perform, we know that we get an extended depolarization for those muscle cells. We know they want to contract, and they want to continually contract to be able to generate enough muscle tension. Now, what we want to start to look at next is combine all those cells, all those muscle cells, and now that we understand the action potentials, we want to see how those muscles work together as a tissue. And if you were to take a look at the muscle layers surrounding the heart, you can see that they're actually all bound together. There's a pattern to them. And that pattern is called a functional syncytium. And here you can see the term sync for synchronization and idiom, whatever that means. Now, the way these cells work, these muscle cells, you can see where they wrap around the most. You can see that they wrap around each of the atria. And then you can see this next band of heart cells that wrap around the ventricles. And what you can imagine, when these cells fire, or when these tissues, when all the individual cells fire at the same time due to the pacemaker cells, you'll see that these will be the first to beat, and then the ventricles are going to be the last to beat. And that's all due to the delay as that pathway travels down that conduction path. So let me draw in yellow. Here I can draw the SA node. I can then move down the AV node and draw that pattern up on this image. So you can see where this pathway is leading. We want first the atria to contract, then we want that pathway to travel down to the ventricles, we want the squeezing to take place down over at the bottom by the apex, and then towards the lateral sides. And the purpose, like I said, the first portion of that muscle syncytium is the atrial muscles. You want the atrial muscles to fire first. That's going to allow enough squeezing of the atria to make it down to the next chamber, which are going to be our ventricles. So squeeze here first, relax the ventricular muscles. That will allow contraction, emptying of the atria, and then filling of the ventricles. Then the next portion, once the atria are going to relax, the ventricles start. So once that's going to make that conduction pathway, makes it all the way down, we're going to get the ventricles to squeeze first. You'll see the first place to squeeze first is down at the very bottom of the interventricular septum, the apex of the heart. We want the squeezing to start from the bottom in order to squeeze towards the lateral sides. Why do we want that to first squeeze out? Take a look at where the outputs are. Okay. The pulmonary valve as well as the aorta. And this is aorta is back over. Okay, I'm sorry, I'm looking at the wrong way. Aorta back over here, so this actually technically goes back underneath. And you see the outputs are towards the top of the heart itself. So we want to squeeze from the bottom and squeeze towards the very top. It's kind of like toothpaste. If you ever see how people squeeze toothpaste, a majority of people that squeeze toothpaste squeeze right in the middle of the tube, right? And what does that do for you? Especially when you're like halfway through the toothpaste. You're just squeezing toothpaste down to the bottom of the tube. That's not the way to do it. You gotta roll it from the bottom and squeeze all the way up, right? So that's exactly what we're doing with the ventricles. Start from the bottom, squeeze all the way up to be able to eject blood efficiently out to the top of the heart. Again, the output being the aorta and the pulmonary trunk. So now that you see the patterns, atria first, then ventricles, we want to break down and take a look at what's taking place, because there's actually 3 different phases that take place. We want to see the atrial contraction take place first. While the ventricles are relaxed, we want the atria to squeeze. That's going to give us enough time to empty out the atria, to squeeze blood and allow filling of the ventricles. Now, when the next phase, as we move down the conduction pathway, we want the atria to relax, and we know when it's relaxing, it's starting to repolarize. We want to depolarize and cause contraction of the ventricles. And as the ventricles, you're going to see there's different phases of ventricular, and it's not called ventricular ejection, it's called ventricular contraction. The first part, just like toothpaste, you want to be able to generate enough tension to build up pressure on that liquid on the inside. You want it to get enough pressure to be able to build up so that by the time you build up enough pressure, it's going to be exceeding the pressure in the aorta as well as the pulmonary trunk. Okay? That pressure is the diastolic pressure, and I'll go into that especially in our vessel lecture. There's a certain amount of pressure because there's already fluid filled up in here. So I want to be able to generate enough tension first, and then when I'm squeezing even harder, that's going to push blood finally to escape. So if I were to give you an example, that would be the door. And that door, imagine this were the heart, and out there would be the aorta. The aorta has enough blood in there, there's enough pressure in there that's about 80 millimeters of mercury. And that, just think of that as a force. We'll just say, for lack of better terms, 80 newtons of force pressing against that door. In order for us to be able to, we're getting filled up by the atria, we're now filling up in here, and we're starting to contract the room. We're going to start contracting the room enough to generate tension so that we start building up pressure. We start off at about 30, 40, 50 millimeters of pressure. We keep generating enough pressure. There's no changes in the volume of blood. It's still all in here. We're just trying to contract the walls enough that eventually we get up to about 79, 80 millimeters of mercury. And the pressure outside is now pressure equalized to the pressure on the inside. We're going to keep contracting. And eventually, when I overcome that 80 newtons of force exerted by the aorta, I can finally open up that valve and now start emptying blood out. So you'll see 2 different patterns when we talk about ventricular contraction or ventricular systole. That's going to be called the, I'll go into it later, it's going to be our isovolumetric contraction and our ventricular ejection. So that's going to be our second phase. Atrial first, atrial contraction, ventricular relaxation. The next phase would be a combination of atrial relaxation and ventricular contraction. And then the next phase after that will be relaxation of the atria and ventricles at the same time. So those are the 3 phases that I will go over on the next couple slides. Given all that, what questions do we have so far? We good? All right. Let's move on with a video. So now this is going to place it all together, because I know my hand animations weren't exactly the best representation of the heart. We've got a video for that that'll play it all for you.
The heart is a muscle about the size of your fist. It lies behind and to the left of your breastbone or sternum. The purpose of the heart is to pump blood through blood vessels, arteries, and veins to all parts of your body. The inside of the heart is divided into 4 chambers. The top 2 chambers are called the atria and are collection chambers for blood. The bottom 2 chambers are called the ventricles and receive the blood from the atria and pump it to the lungs and the body. The chambers are separated by valves, which control the direction of blood flow. There are 4 valves: tricuspid, pulmonic, mitral, and aortic. Circulation begins at the right side of the heart, where blood from the body comes to the right atrium. This blood passes to the right ventricle, where it is pumped to the lungs to receive oxygen. Once it receives oxygen, it flows to the left atrium, and then to the left ventricle, where it is pumped to the aorta and the rest of the body. On the right side of the heart, the tricuspid valve separates the right atrium and the right ventricle, allowing blood to enter the ventricle, but not flow backwards to the atrium. Blood flows through the pulmonic valve to go to the lungs. On the left side of the heart, the mitral valve separates the left atrium and the left ventricle. Blood flows from the left ventricle to the aorta through the aortic valve and to the rest of the body. Arteries carry blood with oxygen and other nutrients throughout the body. Veins take blood back to the heart, which pumps it to the lungs to be oxygenated. The heart arteries, coronary arteries, provide oxygen and nutrients to the heart muscle. The right coronary artery supplies blood to the bottom and the back of the heart. The left coronary artery splits into 2 vessels. One branch supplies blood to the front of the heart. The other branch delivers blood to the left side of the heart. An electric system transmits signals throughout the heart to control its pumping. The electrical signal starts in the sinoatrial, or SA node, which is located in the upper portion of the right atrium and is known as the natural pacemaker of the heart. The electrical signal passes down to the lower chambers of the heart via the atrioventricular, or AV node, which controls the signals so the atria contract before the ventricles. In the ventricles, pathways carry the signal throughout the muscle so that they contract at the same time to pump blood to the lungs and through the body.
So hopefully that pieces it all together. And we'll move over to the next slide, which is going to tie it all together. We want to take a look at the electrical activity that is taking place on the next slide over. So looking at this, you're going to see the autorhythmic, again, our pacemaker cells, in contrast with our cardiac muscle cells action potential. So what we're going to first look at is our SA node, which we know is going to be our pacemaker, so they'll label these P for pacemaker. Our AV node, which is also going to be consisting of our pacemakers. And if you were to look at the waveforms of the pacemakers, here you can clearly see this is the pacemaker potential, here's the pacemaker potential. Eventually we'll reach threshold, and that will result in calcium influx, potassium efflux, and back to where we started. If you were to take a look at the SA node versus the AV node, you can see that the SA node starts, the wave of depolarization is going to start off at 0 seconds, which is going to be the very first starting point. The AV node, you can see taking place about halfway through, so about 50 milliseconds difference in between the first and the second, between the SA node and the AV node. And that represents the delay. And that delay, again, is from the SA node to the AV node, allows for a good enough time so the atria can contract. That's exactly why we want that delay in place. We want to give it enough time so that the atria can fully contract before the ventricles start their depolarization. That's going to describe our SA node to AV node. The next thing is to take a look at, I'm going to use green for this one, the SA node to the atrial muscles. So the SA node is going to be the first to fire. And that is going to trigger all the adjacent atrial muscle cells to fire as well. And again, the different waveforms you would see are atrial muscle, and you'll see our ventricular muscle. Again, rapid depolarization due to sodium, extended depolarization due to calcium, and then continued repolarization due to potassium efflux. So you can see that this pattern right here, the length, which is even more pronounced for A little less pronounced for the atria, a little more pronounced for the ventricles. You can see that the length of the contraction for the atrial muscles versus the length of the contraction for the ventricles. Again, that what I've drawn out represents systole, the length of contraction. And again, systole is everything from depolarization to repolarization. And this should make sense. If you take a look at the atrial contraction, we just want to contract enough to be able to take that blood that's held in the atria and dump that into the ventricles. When we get into ventricular systole, you'll see ventricular systole represents a longer contraction period. Why? Because we've got more blood to move out of the ventricles. So we want an extended contraction period for that portion. And you'll see that these two sections do not overlap. So atrial contraction takes place first, atrial systole, and then ventricular systole takes place after. So not only can you see the node from SA to AV node, you can see the delay in the conduction from SA to AV node. And as this makes its way over, you'll see that SA node, atrial muscle, AV node, ventricular contraction takes place in sequential order as it moves down that conduction pathway.
You said systole is depolarization and repolarization?
The time in between depolarization and repolarization. So if I were to take you back over to this period right here, depolarization to about repolarization. And I know there's a little overlap with repolarization, but you can still just appreciate the fact that once you get to a certain point of repolarization is when the muscle starts to relax. So it is a little bit overlap. But systole is, for all intents and purposes, start of depolarization to the end of depolarization. Probably depolarization to repolarization. That's what I'll label it as. Way too fast. All right, so that is systole. Again, atrial systole, ventricular systole. The other thing that we can look at is the delay in between. So everything out from this point out is diastole. And we want diastole for the ventricles, ventricular diastole. There is an atrial systole and an atrial diastole. There's a ventricular systole and diastole. And all that systole means contraction, relaxation. So don't confuse the two. Ventricular diastole is for the relaxation in order to allow filling. So while this is contracting, the atrial muscles are contracting, that's continued filling of the ventricles. And then once we get to repolarization, we're going to relax the atrial muscles. And then once the AV node makes its way down, or the electrical pathway makes its way down, all the way down to the Purkinje fibers, then we start to see the start of ventricular contraction, or ventricular systole. And this is going to empty blood out to the outputs of the heart, aorta as well as the pulmonary trunk. And then from that point on, we relax. From here to the very end, before we start to initiate the next beat, you can see this period is relaxation. So we can divide that up. From ventricular relaxation, ventricular contraction, and then back to ventricular relaxation. So 3 periods that we can see from the very start of the cycle to the very end of the cycle. And then later on, we'll break this ventricular contraction down, just like I described earlier. Now that we've been able to piece together the action potentials, let me ask you guys, what questions do you guys have? Everybody good? So what we're looking at, just to sum up everything that we've looked at, is one cardiac cycle. And the length of the cardiac cycle, again, really a good chunk of it is dependent on that pacemaker potential. When that SA node fires, you're going to see everything else will fire. atrial muscles, the AV node, as it makes its way down, you're lighting off that fuse that will make its way all the way down and cause the muscles to contract. Now, as I mentioned earlier, this doesn't involve central nervous stimulation. Give me a second. You know what, let's take a break. How about that? Let's take a break. Before I get into autonomic regulation, I'll give you guys 5 minutes, 10 minutes? 7? Okay. 7 minutes. We'll continue at 8:20.